Good afternoon, everyone. Yes, people are still coming, but we need to get started. So it's a real pleasure to introduce Susan Stepney, which probably she does not need any introductions to this community, but will still do this, at least because this is my job. Susan is professor of computer science at the University of York currently and director of the York Cross-Disciplinary Center for System Analysis. She started her career with a PhD at the University of Cambridge in, I guess, in astronomy, right? This was or something related to astronomy. Then she was postdoc in Cambridge. She was a research scientist at GEC Marconi Research Center for a while, and then she took a job at Logica in UK before eventually ended up at the University of York as professor. She is of course very well known in the artificial life community because she also is a co-editor in chief of the Journal of Artificial Life, and of course she's a good friend of ECLT, and so we are happy to have her with us. Thank you, Susan, for having accepted our invitation. So we all look forward to your talk.
Okay, well, thank you very much for inviting me to give this talk. And so what I'm going to be talking about today is some experiments we've been doing over the last, well, several years now on using our automata chemistry to experiment on various evolutionary processes, including replication and parasites. So the work we've been doing has been going on, I think, since actually 2011, and it's been variously funded by the UK's EPSRC and the EU on the EVO project back in the day. So that's it. The automata chemistry I'm going to be talking about is Stringmol, and this is something we've been developing for a while now. So Stringmol, as an automata chemistry, it's each molecule in the chemistry is a little string of assembly language instructions. So it's a string of opcodes, an especially designed language. Now there are 7 functional opcodes which do things like manipulate pointers, copy from strings, read from strings, and then chop strings up. And the one thing that makes it useful for doing evolutionary experiments is that the copy opcode has a very small probability of mutation on copy. So it will copy a character, and with a small probability we'll put the wrong character in place, or chop a character out or put an extra one in. And that's the mutation property. It's a micromutation. There's also a bunch of non-functional opcodes, which are just alphabetic symbols, and we use these primarily to define binding sites and control flow, beginning and end. Now, the thing that makes this automata chemistry rather different from many is that execution occurs between 2 bound molecules. Other automata chemistries that you might have come across, such as Tierra and Davida and so on, it's a single molecule that does its thing by itself. But in Stringmol, molecules bind together and the execution happens when molecules are bound. And this design principle was chosen to sort of model the way proteins bind to DNA or RNAs bind to each other and so on. So one bound molecule executes its program and using the other as data, or at least that's how it starts off until evolution takes over, and then it gets more complicated. And we use a Smith-Waterman-like complementary bindings approach between the 2 strings. As well as the copy to provide opportunities for mutation, there's also, all molecules have a very small chance of decaying. And that means that in order for a particular species to survive, it has to replicate. Otherwise, it will just decay away. So here's a particular Stringmol. I'm not going to go through and say exactly how it works. It's rather complicated because it is just an assembly language. But this is a particular hand-designed molecule that is a replicator. what happens is if this binds to another molecule, it will copy that other molecule. And if it binds to a copy of itself, it takes about 240 time steps to replicate. And so what happens is that you start with A plus A and you end up with A plus A plus A. And what we did in the original examples was we started with a well-mixed bucket of 400 of these seed replicases and watched what happens. And there's quite a lot of complex stuff that does happen. There's a lot of emergent behaviours that get evolved. First of all, there's something we called a sweep, where a mutant species takes over and becomes the dominant species in the bucket. We have a variety of hypercycles that evolved and emerged, and that's where A copies B and B copies A, but neither string can bind to itself, so it can't copy itself. So there's a mutualism that goes on. The only way that A and B can replicate is by copying each other. And in fact, some runs we've noticed multi-species hypercycles with more than 2 interdependent species like this. We also noticed the emergence of macro mutations. So I said the only mutation that is hard-coded in is a small probability of error on copy. But macro mutations emerge where a little bit of functionality evolves that can actually chop strings up and do other quite large changes to the strings. But the thing that I'm going to be talking about in the first half of this talk is the parasites. So a parasite, started off defining it as a species that can be copied, but it itself cannot copy any other species. And the reason for that is it's often lost its copy code, and so that makes it typically much shorter than the other strings, and therefore it gets copied faster, which gives it an evolutionary advantage. But a problem with parasites, certainly in the initial experiments, is it can lead to something called a death spike if that sweeps the population and there's no replicators left. So a little example of one particular run of this well-mixed bucket to show what's going on. Each line is a particular species. And so in things labeled A, we see sweeps. So we had this red species and then it got taken over by this next species, which then got taken over by this one, and so on and so on. Some of these sweeps can be slower. You notice the A sweeps are very fast. There's a quick takeover, but you can have a slower takeover. Then in D, we see the multi-species hypercycles. These are the mutual copying each other, but not themselves. And we can also, during these events, have other species that are sitting around coexisting, so a subpopulation. In this particular run, the hypercycles ended and we went back to replicators and these slow sweeps. And the very final sweep, this one here, was a takeover by parasites. But you can notice this is the last replicator, and when that died out, there was nothing there to replicate the parasites, so the parasites died out and the bucket died. So we wanted to look at these parasites in more detail because there was something interesting going on, and can the strings evolve defenses against the parasites? So that's what we've been doing most recently. In a variant of Stringmol called spatial Stringmol. So we found that these parasites always emerge, and the problem is in the well-mixed bucket, they sweep the entire system, resulting in this death spike. So the hypothesis is in a spatial environment, because the parasites will be localized, the resultant death spike is localized, and so maybe the system as a whole can survive. Parts of it will die out in places, but other parts will survive and be able to repopulate those places. So that's what we were investigating. So here's a picture of one particular run. The color here indicates the length of the strings, and the space is just the space. So very blue strings are very short and are potentially parasites. And the longer mid-blue, yellow, orange, and red strings are longer, and they're the replicators. So you can see when you start off, this is at after quite close to the beginning, after 90,000 time steps, we've still got quite a lot of long strings, these red and orange ones, when we've got blue strings around the edge, which are the parasites. And so the parasites are nibbling away at the edge, but the interior of the region is being protected from the parasites. As time goes on, so we're now 6 million time steps in, we've still got the blue parasites around the edge. The strings in the middle have got a bit shorter. They've managed to evolve themselves to be shorter and have the evolutionary advantage, therefore being copied faster, but they are still replicators. And then time goes on, we're a little bit further on. The strings now have got a bit longer. And I'll come on to that. The reason they've got longer is that they're evolving some defenses against the parasites. And they go on and they get longer and they get more complex. And eventually here, at, that should be 15 million time steps, but there seems to be oh no, so that's that is 1.5 million. This one should be, this is 2 million, sorry. At this point here, after 1.5 million time steps, essentially the parasites have almost died out and the whole arena is full of replicating strings. The defenses have got so good, they have smashed all the parasites and there's no space for them. But then later on, the parasites re-emerge and the strings get more complex again. That's just to show that over a long period of time, you get a lot of complexity, but things do seem to be surviving and we're not getting the inevitable death spike. So here's just a little bit of picture to show the complexity, which is this is time along here, and this is the number of individuals in each species here as a stacked plot. So you can see at certain times we will have dominant species like this blue one here or this orange one here. At this region here, which was the region where the parasites have nearly died out, we see there's a very complex structure of lots and lots of different species, but these are replicators mainly. And then after that dies out, we get other dominant species, but we never get this death spike, or at least we haven't by 2 million time steps. So we wanted to do some analysis of this to find out what actually is going on at this point. So here in this picture, the line along the middle is the string that is the active string, the one that's doing the copying. And the line on the top and the bottom is what it's bound to, what it's copying. Okay, so very close to the beginning time, you have a replicator here. It binds at this point to this string here. And the black line indicates the control flow of the program as it executes. So it starts executing here at the beginning of the binding site. It just walks along the string, doing some various bits and pieces until it gets to this pink region. And this pink region is the active code. This is the copy code. And the line has turned blue because there's a loop here. This is a copy loop. And this copy code is accessing this string and just copying it. And once it's finished copying this string, it falls out of the copy loop and carries on and does a little bit more housekeeping and tidying up until it stops executing here. And in this particular example of this string, it takes 230 steps and you start off with R plus R and you end up with a third replicator, a copy of this string. But this is just a fairly naive replicator. So if it binds to a different string, and this is a parasite string, so it binds to here, it does exactly the same thing. It walks along, it does a little bit of housekeeping, it gets to the copy loop, and this copy loop loops along this string, copying it, and then it finishes off. And so the reaction that you've had is you start with a replicator and a parasite, and you end up with a replicator, a parasite, and a copy of that parasite. And notice this only takes 188 time steps as opposed to the longer one. So in some sense, the parasite has an advantage. It gets copied faster. So how does this then evolve defenses? Well, later on, what's happened, well, you'll notice straight away that the replicator's got a bit shorter. It's evolved away some of its junk that it doesn't need. And what happens now is it now binds to this copy of itself, and it does some stuff at the beginning, not important, but it then moves along until it reaches this particular opcode. And this particular opcode throws the instruction pointer onto the other string. And it toddles along the other string in exactly the same way as it did on the first string until it gets to, hey, that opcode that toggles it on back onto this string, toggles it back down to where it was before. It goes along until it reaches the copy loop, at which point it copies that string, and you get the third replicator. Now, if it goes onto the parasite string, it binds to the parasite string, it goes along until it gets to this opcode, which throws it onto the parasite string. Then it goes onto the parasite string and it never reaches the opcode that throws it back. It gets to something on the parasite string that does something, but it doesn't actually copy anything. It just sort of spins its wheels and then it goes on. So you started off with a replicator and a parasite, and you end up with a replicator and a parasite. So the replicator is not copying the parasite, so you don't get more parasites. But an interesting point is it still takes 218 time steps, which is longer than it took to actually copy the previous parasite. So the parasite not only does not get copied, but it gets caught up in this reaction, so it can't go around and parasitize anything else either. But what happens then is that this another parasite evolves that has this particular opcode in it, and so it passes the check and it can get copied. And so a parasite evolves a defense against the defense. So eventually, by the end, this is what's going on. By the end of the 2 million time steps, the replicator, when it binds to a copy of itself, it goes along, it reaches this point, which checks, and it goes back to this point, but then it reaches another point, which then it goes and checks and goes backwards and forwards until eventually it goes back to this one and copies this string. And it still only takes 200 odd steps to do the copy. And now, with all this extra complexity, the parasite has got 2 of the defense mechanisms. It's got this check and it's got this check, but it still hasn't got the third check, so it doesn't get copied. And it's sitting around bound to the replicator for 191 time steps and not getting itself copied. So you get this evolving complexity of defense and counter defense and defense against the counter defense and so on. So what's happening here is that the parasites are evolving by removing their replication code, which is making them shorter so that they can get copied faster. And one of the ways that the replicators then defend against this, there are a variety of different defense mechanisms, but the main way is to include some kind of check code in their own code that checks whether the partner that they're bound to is similar to themselves. So they don't sort of go and look at the entire code. There's just a little bit of checking going on. Is the partner similar enough to myself? And that stops the parasites being able to be copied. But what happens is then that new parasites re-evolve from these new, more complex replicators, keeping that new check code in them, but still removing the replication code so that they can now parasitize again and be copied and so on. And then more checks evolve and so on and so on. Now, interestingly, what we don't see here is 2 parallel strands of evolution with a strand of replicators evolving from previous replicators and a parallel strand of parasites evolving from previous parasites. What we see is one evolutionary strand of replicators with parasites continually evolving from them, the replicators evolving defenses against those parasites, which then die out, but a new set of parasites evolve from the new replicators so that they can incorporate their code and so on. Well, while we were doing this, we discovered actually that we thought we'd understood what we were doing to start with, but once evolution stepped in, things got a lot more complicated and we had to start being a lot more careful in how we defined things. And in fact, it was very difficult even to define what we meant by a replicator or what we meant by a parasite. We started off with a replicator copies other strings, it copies the string it binds to. But actually, the behavior of the replicator, as we've seen, depends on what the other string is. Once it's evolved from defenses, it will copy nice strings, but is trying not to copy parasitic strings. So it's not just a replicator, it's context dependent. And in fact, given the way the instruction pointer bops around between the different strings, it's not even clear which string is doing the copying, given the complexity. And sometimes the string might destroy itself in the process. So it might make a copy, but destroy itself. So it's not really replicated anything. And there's lots of other side cases and so on. And then also we started off with a definition: a parasite does not copy other strings. That's what makes it parasitic. It can be copied, but it doesn't copy in return. This is even in the simplest case quite difficult, because that's a counterfactual property. We're defining it by something that it does not do. And so it's unobservable in a running experiment because it's not doing it, but it might, we can't tell whether it's not doing it because it's a parasite or it's not doing it for some other reason. And again, its behavior depends on the other string. It can parasitize a naive replicator, but it can't parasitize a replicator that's got a defense against it. It might even replicate some strings and not others. It might even replicate itself, but not the other string. So there's lots, again, of complexity in that. So we ended up coming up with a formal definition of these variety of things. And it ended up that it wasn't a definition of what a string was, because it's context dependent. It's not, and it's not even just a reaction, it's a reaction network, because we have to define maybe 2 or 3 reactions that either happen or don't happen in order to say whether something is replicating or parasitic in a particular context. And formulating these definitions then becomes key to what analysis we can do, because we use the formal definitions to implement our analysis tools based on those definitions. And so we can track how things such as the pairwise interactions affect parasitism and so on, without manually having to step through the reactions. When we first started doing this, there was a lot of manual examination going on of what on earth is happening here. But eventually, obviously, we wanted to automate this, and we automated it based on the formal definitions of the various reactions. So here's a very simple analysis, but I want to say something about even simple analyses are potentially more complex than you think. We're simply here visualizing the length of the strings in the arena as a function of time. So again, we've got time going along here, and we've got string length going up here. Now, of course, given we've got a population of strings in the arena at any time, there's a range of string lengths. So the top graph shows the median string length, and then a few different I think there's a quartile and a 50%, and then a 90% percentile plot. And so you can see, well, there's some changes going on, and you can see that there's this interesting slow decrease in the median length of the string. But if instead of plotting it as statistics as quartiles and percentiles, you actually plot it as a heat map of the actual lengths of the string, you see there's a lot of structure in the string length distribution at any time that is not visible if you just plot these medians and quartiles. And in particular, you see here the slow decrease, but that you can see that there's also other subpopulations of strings of different lengths, which are also undergoing similar slow decreases in length. So the slow decrease in length gives you some slight evolutionary advantage because you get replicated faster, but you can see that there's a general theme, but you also get these bigger subpopulations as well. So these plots are much more useful for spotting interesting things happening than these kind of summary statistic plots. Here's another plot. Now, the top is the run that I've been talking about all the time, and the bottom is a different run, just to show another difference that happened. So here, the color coding shows the number of strings in the population that are undergoing a particular class of reaction. So the red ones are non-replication reactions. They are reactions the strings bind and do something but don't replicate. Green are hypercycles, orange is a mutual or a self-replication reaction, and a blue is a parasitic reaction. So here we see the blue parasites, they never get really dominant. There's this region here where they nearly die out, and then they come back again, but still don't get very dominant. And the reason I showed this other run is there seems to be a completely different mechanism going on in this other run, is that hypercycles evolve here. And remember, a hypercycle is where A can replicate B and B can replicate A, but they can't replicate themselves. So they need to have both A and B present in order for this to happen. And somehow this seems to protect against parasites, possibly because the parasite can get a defense against one of the replications, but not the other, maybe. We don't know. That's something that we're going to be looking at next. So the lessons on the parasite side, this simple replicate Stringmol, it's not particularly complicated. We start off with just a bucket or an arena of identical replicas. Exhibits very complex behaviors through this very simple small probability of mutation on copy of a single symbol. And so we get complex behaviors, hypercycles, parasitism, and so on. And in order to stop the parasites just killing everything, we really do need space. This has been found in other systems, and we've demonstrated the same in our system with the more complex evolutionary characteristics going on. And it does appear to be that the parasites are the drivers of increasing complexity in our system, as well as being hypothesized in other systems. Shorter strings are copied faster, so they have some evolutionary advantage, but longer strings can include more complex defenses against the parasites. And it seems to be that this complexity wins out in the end. And we see this because in that case where the parasites need to go extinct because the defenses are so good, the complexity of the remaining replicator strings decreases. It evolves out its defenses because, of course, why wouldn't it? That is just extra code that is making it slower to replicate. But once it's evolved out its defenses, the parasites rise again, and then the complexity increases again as the replicators evolve defenses against this new range of parasites. And also, we learned some lessons doing the analysis. The summary statistics are not particularly helpful. They don't show the fine detail. They don't show the edge cases where potentially the interesting behaviors are going on. Also, there's a lot of generic measures in the literature that are suggested are going to be useful for comparison, but actually they're not particularly useful because they make a lot of assumptions about the system. So I said right at the beginning that Stringmol is different from things like Avida and Tierra because it requires two strings to bind. And so a lot of assumptions that some of the generic measures make don't take that into consideration. So in fact, our analysis followed an experimental approach. We observe the system, we hypothesize, we observe something weird happening, we hypothesize what's going on, we develop some tools to measure and test that hypothesis, and then we go around that loop again as we get more and more sophisticated analyses and better understanding of what's going on. But also it's behavioral measures that are key. The innovations and transformations that we see in the happening in the system are behavioral rather than structural innovations, and behaviors are very system specific. For example, this complex evolved flow of control that's happening in the Stringmol, and you can't see that that's going to happen from a static analysis. You actually have to execute and watch what's actually happening. Okay, so that was a brief summary of the stuff that we've been doing with a simple Stringmol system. I now, just in the second half, want to go through a different set of experiments that we've done with Stringmol a while back, which is looking at the von Neumann universal constructor architecture and seeing what happens when we try and implement and evolve that in Stringmol. So just to remind people, this is the schematic of the universal constructor architecture. We've got two parts to it. We've got the static data and code, which is this orange part, which is analogous to the genome. And here, say, it's got four parts to it. Part of it is the code for copying, and what the copier does is copy the genome. Part of it is controller code, which is just mechanisms for saying when things are copying and whatever. Part of it is expressor code, which is to apply to the genome in order to make the little machines that are actually the active bits of code. And then there's any other application code or payload or whatever you want to call it that is around. And these active machines, which get expressed from here, we can call these analogs of the proteins. So we have a copy of protein or ribosome or whatever you want, but that's why proteins are in quotes. Then a controller protein, an expressor protein, and so on. And that's the high-level architecture of the system. So first of all, we started off with a fairly naive Stringmol implementation. We'd had experience with writing replicators and so on. So what we started off doing was say, oh, okay, the proteins are just little Stringmols that do whatever it is they've been programmed to do. And the genome is just a string of the relevant Stringmol machines. Expression is therefore trivial. It is just chopping up the genome into the relevant proteins. But the problem with this implementation is there's actually no way to distinguish the static genome, what's supposed to be the static genome, from the active proteins. And in particular, the copiers bind to too much. They bind to everything. And there's no way to constrain them to copy only the DNA. They also copy the proteins. And because they copy the proteins, the DNA really has nothing to do, and so it evolves away. So the original structure evolves away, just leaving all a bucket full of the separate protein machines. So that wasn't very good. That wasn't what we wanted to happen. So we had to think about that a bit more. So we ended up with this instead, a DNA-like encoding, which is a little bit more complicated. Rather than the few hundred opcodes we've seen before, we've now got 3,500 opcodes in the DNA, which is a non-trivial encoding of the proteins into the genome. So G stands for genome and 0 stands for the initial genome, because we're computer scientists, so we start counting from 0. If you're unhappy with that, pretend it's a 1. Then what we have is non-trivial expression now. So the expressed machine looks like this. So that's hiding inside this very long genome, encoded in the long genome. And taking the inspiration from DNA, there are complementary triplets in here, which are translated by other information in here into a single Stringmol. So we can think of this as the DNA with a lot of structure in it, and we can think of this as now our active protein Stringmol, which has got code in it to do stuff. And then the copier, the expressed copier, looks like this, and it is designed such that it will copy only the genome and it won't copy the proteins. And so it's got a complicated structure. But this structure maintains while evolving. And all credit to my co-authors who did the work. This is hand designed, this is hand designed, and the encoding is hand designed, and that's the starting point of the system. If anybody wants to go and hand design a different thing, good luck to you. You need to be into Stringmol to understand how to do this. But okay, so this is the normal operation of the copier. So this is just a kind of a flow chart. The time runs down. The black line represents the place where the two strings are bound and are reacting. So here we start off with the initial copier. So this is the protein floating around in the system, and the initial genome floating around in the system. So the copier binds to the genome, and what it makes is another copy of the whole genome, and then the binding stops, and so the original genome and the original copier are restored as well. So you start off with a C and a G, and you end up with the C, the G, and a copy of the G. So that's what it's designed to do. And this is the normal operation of the expressor. So you start off with an expressor protein floating around in the system, and it binds to a genome floating around. And its job is to produce a copier, an expressor, and the payload. So there are basically three machines encoded into the genome: the copy machine, the expressor machine, and the payload machine. Okay, so that's what the expressor is supposed to do. So that's the entire design of what goes on. But remember what I said, like in the other Stringmol, there's a small chance of mutation on copy. So when things are, when both these machines, the copier and the expressor, have to do some low-level copying, there's a small chance of error on that. So let's have a look at a variety of things that happen here. Sorry, this one's a bit busy, but hopefully you can see what's going on. This is a copier and a genome come together, and there's the copier is supposed to be copying the genome, but there's a small error on copy, and it actually produces a mutant genome, this G1, pink one. Then the original expressor binds to that mutant genome, and it expresses, well, it expresses the original copier, but it expresses a mutant expressor, and then it expresses the payload as well. And then interestingly, what happens is that this mutant expressor binds to the original genome, and it produces a mutant copier, another mutant expressor, and the payload. And then this second mutant expressor binds to the mutant genome and produces the same mutant copier, yet another mutant expressor, and the payload. And then this final mutant expressor, E3, binds to the mutant genome and produces the mutant copier, the mutant expressor, and the payload. So by this stage, we are now back to a stable state. And these graphs show the time variation of, here's the copier, the original copier dies out and the new copier comes in. With the expressors, the original one dies out, the mutant comes in, but then dies out as the final mutant comes in. And then the genome, the original genome dies out and the mutant genome comes in, and the payload actually never ended up in this particular example of being mutated. So that's kind of what we had hoped to see when we designed this system. We had hoped to see evolution, and evolution of not just the payload, which other authors have actually managed to find as well, but here, evolution of the expressor and copier parts as well. So the expressor and the copier have also changed. And other authors prior to this have found that their systems, if the expressor and the copier changed, unfortunately that damaged the system too much, and you ended up with non-viable expressors or copiers, but here they've stayed viable. But that wasn't the only thing that happened. Here's another thing that we noticed, which we called semantic takeover of the expressor. So here we have the expressor and the genome, and the copier and the genome, and the copier makes an error and it makes a mutant genome, G1. And then the expressor comes in and binds to this mutant genome, produces the original copier, but a mutant expressor and the payload. That mutant expressor now binds to the original genome, producing the original copier, yet another mutant expressor and the payload. And that final mutant expressor binds to the original genome and produces the original copier, a copy of the same mutant expressor, and in this particular case, a mutated payload as well. And so this has now stabilized again. And what I want you to notice here, so here again, we have the takeover of expressor 1 by expressor 2, and the takeover of payload 1 from payload 0. But what I want you to notice here is that the genome is essentially now the original genome. There was a transient mutant genome, but that transient mutant genome never survived. And in fact, was so low numbers, it's never appeared on this graph. And so at the end, we end up with a different expressor, but the same genome. Genome has not changed, but what gets expressed has. So that was interesting. And we saw other things here. So it's not just the expressor that can change. This example shows the copier changing as well. So copier and genome, mutant genome, expressor and genome, mutant expressor, mutant and original genome, mutant copier, same expressor, and then that copier now the mutant copier can still copy the original genome. So now we have, the result is original genome, but we now have a different copier and a different expressor being expressed from that genome. And then finally, the one that really amused us, semantic change without any miscopying of the genome. So there's no mutation of the genome happening here at all. Expressor binds to genome, expresses the copier. Now the expressor expresses a different genome by accident. Sorry, the expressor expresses a different expressor by accident. There's a mutation on expression and the same payload. That mutant expressor binds to the original genome, produces yet another mutant, but that final mutant has stabilized and baked in. And so here we start out, we never changed the genome, but what's getting expressed has changed. And so we say that's a semantic change. This genome hasn't ever been changed, but what it expresses has been changed. So just to see the lessons that we learned from that, we can build a Stringmol implementation of the von Neumann architecture, and it too exhibits complex evolutionary behaviors. And so errors on copying can change the genome, as one might expect, and therefore change what it expresses. And these changes can even be baked back into the original genome. They're still visible in the original genome because, of course, the original genome is now in a different context. But errors on expression alone can also result in fixed changes to expression. So in that one, we saw E0 expresses E0 and E2 expresses E2. So which expressor is actually encoded on the genome? Well, it's context dependent. The meaning of the genome is relative to the context it appears in. So just to conclude, the Stringmol automata chemistry exhibits many what we might call lifelike properties, evolution of interesting behaviors such as parasitism and hypercycles, evolution of the von Neumann replicator with interesting changes in expression, and even relative semantic change of what's being encoded in the genome. And in future, we're going to continue working on this to look at more of these kind of open-ended behaviors. And for those of you interested, I've got the references here for the papers that I've been talking about today. So thank you very much.
Thank you, Susan. Thank you very much to you. This is very, very, enlightening, I think. So are there any questions for Susan? Yeah, Rudy has a question, please.
I can't hear you, Rudy. I can't hear you.
Oh, sorry. Now it should work.
Yes.
I think another 2 years and I will have learned. Okay, good. First, Susan, thank you very much for this talk. As I expected, I learned a lot, and it's simply brilliant. Thanks a lot. Now, my question refers to the open-endedness of evolution. What would you say, what was, or I'll ask it deliberately slightly provocative but I don't want to do any harm. Please take it right. What's the most unexpected behavior that you have seen? Or of which you can be sure that it is not implicit, that is not somehow implicit in what you put in, but by, as you said it, hand coding.
Mm-hmm. Yeah, I think possibly one of the more interesting one was the evolution of macro mutations.
Yeah.
So we know that we've hand coded micro mutations, but the only mutation is mutation on copy, and copy of a single symbol. So it's copying the whole string, and so it can copy a single symbol. But some macro mutations evolved that could do larger scale changes to the strings, and these were preserved through replication and mutation as well. And we weren't expecting to see that. We were hoping to see a lot of the other stuff that we saw, like parasites. We were expecting to see parasites because they've appeared in other systems, and we were even expecting that by putting in space we would stop the death spikes. We possibly weren't expecting the hypercycles to be a defense against parasites, and we still don't quite understand what's going on there. Maybe when we understand what's going on there, we will realize we did expect it, but at the moment we don't understand.
Okay. Thank you very much, of course. Well, that's absolutely great. Thanks a lot. I think with respect to hypercycles, John will certainly have many contributions, but I understand now. Macro.
Thank you, Rudy.
Thanks.
There is a question from Martin. Martin, please.
Great. Thank you, Susan. Just first observation from when I was a PhD student working with ribozymes and doing selections on ribozymes. It was true in that case, we're working with a ribozyme of 490 nucleotides, and every generation we had to physically eliminate the shorter variants from the pool because they kept coming back. And we called them, and we did find some of them to be parasitic. So the continual emergence of parasites was also found in these types of systems. My question is, in some of these other systems that you briefly mentioned, where they're looking at evolution of these kinds of systems, for example, self-generating chemistries based on lambda calculus, you probably know those guys, right? So they found also that different kinds of things, maybe like parasites, could drive complexity, but they have to artificially go in and say and prevent some things from happening. For example, that A replicates A. If you don't eliminate the simplest reactions, then the systems never generate complexity. So what about your system? Do you have anything hidden in the background where you try to eliminate it from going just to very simplistic outcomes, or?
Well, I think the only time the elimination was in the von Neumann architecture, where it did simplify itself and we needed to add structure, but that wasn't adding any structure to the code underneath, what we call the physics. That was only in the biology, in the actual structure of the strings. Now, I do admit there are a variety of parameters, such as mutation rates and things like that, and decay rates and so on, which do have to be tweaked such that not everything dies. Because if you decay too fast, everything dies, and if you decay too slowly, there's not enough evolutionary pressure. So, but it's not that sensitive to it. You just have to move it around. But certainly, self-replication, A copying A, is the whole basis of what we're doing. So yeah, as far as I'm aware, unless my co-authors have snuck something in, which I'm pretty sure they haven't, well, we haven't had to do anything like that. It's just been the very careful design of the initial conditions, Great. Which I agree is a careful design.
Yeah, very interesting. Thank you, Susan.
Thank you. Maria, please. There is another question from Maria.
Okay. Yeah, so first of all, thank you so much for your talk. So I'm more a curiosity. So this model, the model of disappearing and reappearing parasite population might be used as some kind of formal tool to encrypt information, because actually that reminded me of dynamics of entanglement in quantum computing and quantum mechanics, because when you have this information that goes down, actually there is no way to retrieve the information, then it goes back. Here you can have a system which is kind of controlled by the environment. So I'm wondering if it could maybe in the future lead to a new system to deliver information.
That's an interesting point, but it does lose information when the parasites kind of die out, and it does have to regain that complexity afterwards, but there's no guarantee it's the same complexity that it regains, because what happens is it loses its defense against the parasites that have died, new parasites emerge, and then it has to evolve more complex behaviors to guard against those new ones. But because it's just evolving sort of randomly, it might be able to evolve, it might the second time around evolve a different new behavior. either because the parasites that have evolved are different, or simply it's come across one of the other kinds of defenses. So I don't think you could guarantee that it's going to remember the information that it lost. It just remembers, it recreates complexity that it needs to survive.
Thank you.
Thank you. John, please. There is another question from John.
Yeah, Susan, thanks for the talk. I guess in 1990, when we started building the computer engine, almost the entire motivation for spending a couple of years building a new type of computer was to simulate our version, I guess, of what now is called Stringmol, which we called polymer chemistry on tape, and got documented a lot in Marcel Turk's thesis back in the late 1980s. So I feel that in some sense it's wonderful to sort of relive the atmosphere of our group seminars back in the late 1980s. But I'm kind of wondering, it looks like that the current tools should enable one to go a lot further on the subject. And so the first question is, with respect to computer timing, I notice you're sort of doing 10.5 million sort of time steps. And with our NGen computer, we were, if the time step is a sort of an individual processing step, we managed to construct things so that the computer clock frequency was the processing step time with our reconfigurable logic implementations of string chemistry, which was a sort of mainly a binary chemistry, such as the same type that you're talking about. But so that the question is, with respect to looking at long time, we were sort of tantalized by getting the number of generations associated with real evolution up in the 10 to the 12th generations. If you sort of take a classic origin of life type picture where you're talking about day-like temperature cycles as the sort of origin of initial replication, then you have to get up to about 10 to the 12th to relive the evolutionary history of the Earth. And that was the motivation for us building our computer back then. So I'm sort of, I'm intrigued at the similarities. And on the other hand, I'm sort of, with the current developments with with GPUs and other things, I'm sort of asking the question in my mind about the time scale at which you're looking at, because a lot of your conclusions seem to be, well, we let it run for a bit and see what happens, but you're exploring a time range which seems much shorter than what could be realized.
Yeah, the original experiments we were doing was sort of just on a workstation. There has been a bit of moving to a cluster, but that's not to speed up an individual experiment. That's to run lots of experiments in parallel, because obviously they're evolutionary, multiple evolutionary runs. I was mainly concentrating on a single run in that case. So one of the things we are potentially looking at is ways of speeding it up. When I say a time step, I simply mean execution of a single opcode. Now, obviously, some of those opcodes are skip, and some of those opcodes are copy this symbol, which is unlikely to take the same actual amount of time. It is just currently an interpreted language, but there have been various speed ups on it. Personally, I think that what is needed more for open-ended evolution experiments and so on is the inclusion of a richer environment. At the moment, we've got a bunch of strings swimming around. I can
second that. Yep.
Yeah. And so there's nothing for them to get any evolutionary advantage off of except each other. We do have ones where we've looked at giving them different amounts of energy, so they need to have an energy in order to execute their opcodes. But given that there's also a time penalty for being big, having an energy penalty as well doesn't seem to do anything new in that particular case. What would be interesting if there was a way, for example, of storing energy. So if there was a lot around when you weren't using it, you could store it and use it later. But I think actually having a richer environment is, I mean, just going from a bucket to space gave such different behaviors. Having some kind of structure on that space, I think would be the way to go next.
And one second question, if I may.
Please.
Is that all right, Achille?
Yes, please, go ahead.
It relates a little bit to Martin's point, but on a slightly different tack, having studied molecular replication systems in the lab. The copy process in Stringmol is comparatively easy process, at least the maybe not the completion of the whole copy process, but the actual copying of the copy operation is a sort of, it's like a quantum element, if I understand things correctly. It doesn't cost a lot to make a copier, a copy operation. So I'm asking the question with respect to Stringmol about the choice of the operations and their facility. Do you think that the basic operations that you provide and their dependence upon the strings in the system have an important impact on the types of phenomena that you're seeing?
Yeah, so we've done some other smaller studies. One was to have a soft copy where actually the copy operator itself was broken down into 3 or 4 stages with different opportunities to go wrong at the different stages, and even to do some backtracking and so on, which produced some a much slower version of Stringmol, but was potentially interesting. But one of the other things we also looked at was the copy operator. There's the symbol that it produces is magically produced from nowhere, and we did a kind of a mass conservation experiment where the strings that decayed formed a pool of symbols, and when the copy operator came along and tried to do a copy, if this copy symbol it needed was in the pool, it would use that, but if it wasn't, it kind of took another one at random out of the pool, and that had some interesting consequences as well. And I think again it's those kind of interesting constraints that are kind of caused by physics or the environment or whatever, that also can produce a lot of interesting complexity and evolutionary pressures that you might not be expecting.
Thank you.
Okay, I don't see any other questions, so please join me in thanking Susan again. And it was a pleasure to have you here, and we hope to have you, with us in the future in person.
Ah, that would be nice. Yes. Thank you very much.
Thank you.